Hello, it's your boy, the one and only Sir Patrick of the Department of Biology. So I hope you enjoyed that last activity. Now we'll move on to the next activity for this chapter, the use of Mega software. So we'll have a brief introduction on the basics of Mega software and the use of NCBI database and Blast search tool. To give you an idea of how we really do it on the field. So we have contacts on some what do you call this company, sequencing company, laboratory companies, and they will usually send you some order form, or you need to accomplish some order form first. So the order form looks like this. It has, well, the guidelines on what will you do if you want to have a sequence, if you want to sequence something. And sometimes you will need to package your materials, and we use this Eppendorf tubes to actually, yes, this Eppendorf tube to suspend your DNA extract, or sometimes suspend your bacterial isolates. And we cover it with a lot of cling wrap to protect it for the long travel. Kasi lilipad pa yan papuntang Korea. We have a sequencing facilities here in the Philippines, but it usually takes about 10 days for them to sequence and amplify your file, your isolate. So we tend to always use sequencing services from other companies outside the Philippines, like for example, Macrogen in South Korea, or in First Base sequencing facility here in Malaysia. Okay, so simple packaging recommendations. We usually use Eppendorf tubes to contain the DNA pellet, and we suspend it in nuclease-free water or TE buffer. And sometimes we use the isolate itself in agar. We send those isolate in agar. So we scoop some of the isolates and put it in an Eppendorf, and we cover it with a lot of cling wrap and label these Eppendorf tubes. Then we send it to FedEx, and then FedEx to South Korea. Okay, so in the accomplishment of the order form, you will notice there are a lot of universal primers that you can actually opt to use when you want to sequence your isolates. Usually, what I have experienced in using universal primers, well, I have just been doing two things here. We usually use what we call ITS, your internal transcribe spacer. So ITS1 or ITS4. We usually use these two. So the ITS1 is your forward, and ITS4 for the reverse primer. And these two are really good in amplifying for ID your for IDing your isolates, no, especially for fungal isolates. So ITS1 is and ITS4 are used for fungal isolates, no, fungi DNA. But when you want to ID bacterial isolates, we usually use these two primers here. These are for your 16S primer. Okay, so 16S are RNA, no? Both are ribosomal DNA, no? And yes, those are the two, ITS, and for the 16S is 27F and 1492R. Okay, the forward and the reverse. So this is how the order form usually looks like. As you can see here, we have a lot of things to accomplish, but don't worry about it. I can teach you how to doggy. So in here, you can see there are reaction information, sample information, and the primers that you will use, no? As well as the sample type and your service that you want the company to do. So here you can assign a name for your sample. Usually you use the name of the campus, so UPB something, UPB1, UPB2. So be creative in doing this, but don't use too long names, no? Because you need to put it in the Eppendorf tubes as a label. So there you go. As you can see, you need two for each sample or each isolate, bacterial isolate or fungal isolate. You need to put in two, two, two cells, no, two rows. I mean, for the forward and for the reverse primer. And you usually don't have to put in um, something here, no. It's okay to put just Na, and you need to put the sequence of the primer, the universal primer that you will use. For this case, we are using your 27F and 1492R. As for 16S, our DNA application. And you need to put in what type of sample you're using. For this case, we are sending purified and extracted DNA. So what the company just need to do is, instead of extracting, they just need to PCR amplify and PCR clean up and standard bidirectional sequencing of your isolates. So that's it. That's it for this order form. So for this activity, we're going to be using the same rubric. So the content, use of language, organization, and timeliness. So this activity, of course, follows Lecture 7, Molecular Systematics. And as you have remembered from the study guide, we have at least 3 steps in that activity. The first one would be Mega X sequence cleanup and Contig assembly of your bidirectional sequences. Okay? So I hope you find this activity file in our VLE course site. And for the basics of Mega software, we have the part 1, Mega X sequence cleanup and Contig assembly of your bidirectional sequences. So the first step would be to download the DNA trace file. Okay, these are your what we call chromatogram. So go to the VLE core site and find the folder raw sequences. Okay, so it should look like something like this. Let me just find this one. Here, this folder titled raw sequences. So, okay. So you need to look at the file with, yes, this one ending in .abi. So these are your DNA trace file. So the file .abi will be the one that we are going to use. Those are your DNA trace files, okay? Those are the chromatograms. And you need to go to this site. Let me just click it, allow. And then this is for the download of your Mega software, no? Molecular Evolutionary Genetics Analysis. So this is a very good software for this particular part. So it can be used on Mac OS, Windows, and so on. I need you to choose the graphical GUI, and you can choose either 64 or 32-bit depending on your computer. Then hit download. After the download, you can just install the exe file or the DMG file that you have downloaded. So it should look like something like this, okay? Molecular Evolutionary Genetics Analysis. This is your Mega software. Okay? So it has a lot of buttons. Don't worry about it. We'll just use some of it anyway. Okay, so after downloading and installing, you need to watch this video. And now, for some of you who are lost and don't know what's happening, here we're using the ITS1 and ITS4 for our forward and reverse primer in the PCR amplification. Okay? Or in the sequencing of our fungi. So your ITS are, these are your universal primers, okay? Specifically for fungi. So ITS is the internal transcribe spacer, no? So ITS is the internal transcribe spacer. So as you can see, to help you visualize on what's going on at the molecular level, take a look at this, no? So we have your ITS1 here, the forward primer, okay? So this will amplify the downstream, okay, of that region, while your RTS4 will amplify the upstream portion. Something like this, okay? So as you can see here, the forward and reverse primer will somehow meet at the end, no? So we have your 5' end here and 3' end here. So this is how your ITS look like, no? The generic fungal ribosomal gene. Okay? So as you can see here, we have a lot of things going on. Okay? And the important portion of this diagram, this schematic diagram, is that we have the 18S, 5.8S, and 28S regions, which are conserved among fungal species. But the internal transcribed spacer between them, the ITS region between them, are highly variable. And that will allow us to discern among the species of fungi or isolates that we have if we cross-reference it in their database, in the NCBI database. Okay? So that's what's happening. Now, to give you an idea of what's happening here, this is actually a part of my study on the land use practices and occurrence of your entomopathogenic fungi. And I surveyed, or I'm comparing the species that are found in your agricultural patch and Mosiforest patch of your Atok, here in Atok, Benguet, no? So I conducted a lot of soil samples to compare the, well, entomopathogenic or fungal species in the soil of your Mosiforest or forested patch and your agricultural patch. And I use this insect here, this bait insect, Zopobas morio, to compare the fungi, entomopathogenic fungi, or the fungi that can kill insects in these two sites, no, your agricultural and Mosiforest site. So that's how this data come to fruition. It is a part of my study. So this raw sequences file that we have here is actually a part of my studies. And it cost me about 10K just to have this. So let's input this ABI file, this chromatogram file in the Mega software. So you can just drag it there. And as you can see here, it looks like this. No, you can adjust the height of the waves of your chromatogram here, or adjust the distances between the peaks right there. Okay, so you can adjust it, the height here. Okay, pwede yung paglaro-laruan yan, or you can adjust the distances between the peaks here. Okay, so it looks so cool, right? And the numbers are actually important. Numbers above, these are your, well, the upstream to downstream, no, of your DNA sequence. So what are these peaks? These peaks are the reads for your DNA. Okay, so as we all know, there are 4-letter, well, in the nucleotide sequences, there are 4-letter words. I mean, yes, we have the ATGC, remember? And these are the peaks, no? The reads here. Those peak represents that. And they are color coded. That's why you can know which is T, which is G, and so on. So, yes, you can adjust it. And if you look at it, look at the whole file, there is something wrong, no? Especially on the ends here, on the downstream end. Look at this. We have something going on right here on the downstream end, no? And if you look at the upstream too, there is something wrong too, no? So if you look at the first portion, the header, kumbaga, of your sequences and the last portion of your sequences, there is an anomaly, no? It doesn't look quite well as the middle portion of your sequences. So we need to do something about this. There are errors here kasi, no? So, pangit yung mga reads here. Okay? So, what we need to do is we need to mask the sequences. So, you need to choose, as a researcher, no, you need to decide which part you need to keep or which part you are going to mask. So, what we're going to do is we click this portion to mask the upstream portion, and we can also do it on the downstream end too. So you, as a researcher, you decide what to keep. So I'm gonna click T, and I'm going to mask the downstream portion of your sequences, which is super pangit. So yeah, when it's blackened, when it's dark na, it's masked na. So what's that gonna do is it's going to, well, mask or remove the sequences, those unreliable reads in our sequence file, okay? Because those reads, as you can see, are pretty pangit, no? And unreliable. So it might mess up our sequence. So yeah, we need to trim those out of our sequence. Okay, now what I'm gonna do next is export it into a FASTA format. So FASTA, that's okay. And this is what I call cleaning, no? That's why I'm gonna rename it, let me see, UPB CRE2 cleaned, no? Because we have trimmed out or cleaned the sequence, kumbaga. Okay? Okay, now what we're gonna do is do it for all of the other sequences. And if we look at it, so this is now your trimmed and cleaned UPBCRE2 sequence, okay? Now what we're gonna do next is do it for the other ABI file. So let me do it for UPBCRE2 ITS1, and we're now gonna do it with our ITS4 ABI chromatogram file. So let me import it again. So this is how it looks like. Okay, so you need to adjust here so you can move it. So let's now see, as you can see, you can now move it. And yes, just like the ITS1, for UPBCRE ITS4, there are also anomalies. So we're gonna trim out those portions too. So you need to decide amongst yourself which part to trim out. The key here is look for anomalies, no? Yung mga pangit na regions, yun yung i-trim out nyo. Pero wag masyadong i-trim out because it will shorten too much, no? Our file. So I'm gonna rename this UPB CRE2 cleaned ITS 4. Okay? There. Okay. So those are the two clean sequences. So I'm just gonna rename this. So cleaned ITS 1. So it's organized. So you need to do this for all of the ABI file. You need to clean all of those sequences, okay?
One eternity later.
Now that we have cleaned all of those sequences, fast forward na natin, we have this clean sequences file, no? UPBCRE 1 to 5. So yes, so I also changed some of the names. So that's the thing here. You need to be organized so you don't get too cluttered, no? The next thing that we will do with these clean sequences is, wait, let me just rename this folder, okay? Okay, I'm just gonna rename it so we're organized. So cleaned, I'm gonna change it to 0 clean. Okay. So we're numerical. Okay, so what we need to do next is we need to create a new alignment. So click, let me click align, edit build alignment, and yes, edit build alignment, create new alignment, DNA. Okay, so it will look like something like this. Now, click Edit, insert sequence from file, and find your clean sequences. So I have named it zero clean. So where it is, yeah, there it is, zero clean. I'm gonna import that and that too, the forward and the reverse, ITS1 and ITS4. So as you can see, it's now imported. So I'm gonna delete this blank sequence here. So I'm gonna select it and click delete in my keyboard. So it's gone now. So we only have your UPBCRE8 for ITS1 and ITS4, the forward and reverse. You can click this species and abbreviation to, organize them, order them alphabetically. And for the alignment, we have two algorithms that we're using, the cluster W and the muscle algorithm. Primarily for this activity, we're going to use the muscle algorithm. Okay? So, furthermore, you can also delete a certain portion of your sequences if you'd like to do that. You can select it, no? You can select a certain portion. You can add a certain portion, add a letter, no? Letter base, depending on what you like. And you can input something like repeating t t t t t t t t, or you can also input other bases. No, you can edit it any way you like this, and you can undo that edit using Control Z. So yep, I encourage you to play with it so you know how the nitty gritty of the system works. Now moving on, right click the ITS4, the reverse, and click reverse complement. So after creating the reverse complement for ITS4, select both of them. So just like you select files and click align DNA. So muscle align DNA, and just don't change anything here, and click OK. And it will align your sequences. No? So should look like it should look something like this. Okay. So it's now aligned. So what we need to do now is to edit the ITS1 portion. No? So as you can see, we have site number, and I'd like you to select, wait, select, oh my god, okay, here. So select the first portion and paste it on the top portion, something like this. And for the downside, the downstream part, you don't need to do anything. Okay, so it remains the same. So as you can see here, there are differences, but that's okay. Don't worry about them. Those are the errors sometimes. And I'll explain it later why it looks something like that. Okay? So it should look something like that. Now, after pasting the header of your ITS4 to ITS1, you need to delete this. Okay? And let me change the name of UPBCRE8 now to UPBCRE1. Okay, so it's much more organized now. And I'm gonna export it, export alignment FASTA format. So I'm gonna name this UPBCRE1. I have changed the 8 to 1 now. So UPBCRE1 Contig assembly. Okay, and click save. So close that, and you can do it for the next portion. Let's see, for the UPBCRE2 naman. So edit build alignment again, create new alignment, click OK, DNA. Now edit, insert sequence from file, find that file. So desktop zero clean pa rin. So zero clean, UPBCRE2 naman. So both the reverse and forward. Okay. Now delete the blank sequence here. Select it, delete. Okay. And you can arrange it again. So ITS1 and ITS4 reverse complement. Then select them, click muscle align, okay. And then it is aligned again. So we'll edit this one again. So we select this portion, copy it, and paste it right here. So you need to select that specific portion you want to paste on. Okay. Now I have deleted the reverse portion and renamed it to UPBCRE2. Now I'm gonna export it now to FASTA format, and I'm gonna rename it UPBCRE2 Contig assembly. Okay. All right. So that's how it works.
One eternity later.
After doing it for all the clean sequences, we now have the final config assembly for all of our isolates. No? Okay, we're now almost done. So we have UPBCRE1 to UPBCRE5 config assembly. All right? So yep, this is the final product of the first portion, the sequence cleanup and creation of your config assembly from the bidirectional sequences. So why are we doing this anyway? Well, this is an attempt of us to somehow replicate the sequence assembly. Because if you, specifically the naive approach to sequence assembly, if, say for example, I have a DNA sequence, 5 prime to 3 prime, and another sequence, so we have this first read from your ITS1 and second read from your ITS4. So if I have a significant sufficient overlap between the first and the second read, well, I am convinced that the sequences on the second read matches the correct location as on the first read, then I can say that those sequences somehow go together. No, it's like a naive approach in sequence assembly. Then I can say that those sequences is just basically I can generate one continuous config that goes from the prime I prime end of the first and to the 3 prime end of the second. No? So theoretically, this is an easy problem to solve. No? So all I've got to do is I just look at the sequences, the first read and the second read, and I say I can find my 5 prime end and 3 prime end that match and put them together into one continuous config. No? But there are a lot of complications usually in this genome assembly, this naive approach that we're using here. So I'm not actually recommending it, no? Because sometimes we may not always have exactly the right basis. As we have mentioned earlier, there are mismatches, no? These are errors. So I only recommend using this approach um, for some assemblies, no? We don't usually use this naive approach in sequence assembly unless we have a very simple region that we are sequencing and you're just putting together a very short reads. Now that we're done with the first part of our activity, we'll now move on to the use of your BLAST program, your NCBI BLAST. Okay, so for the part 2 of activity 2, NCBI BLAST program, what we need to do is we need to ready our cleaned and assembled contigs, no? So it should be in FASTA format. Now you need to click this website, go to this website, or you could just type in BLAST in Google and it will direct you to this. Now, BLAST is Basic Local Alignment Search Tool. So from the name itself, it is an alignment search tool. No, it is a bioinformatics algorithm or a program that can compare your primary biological sequences, like for your amino acid sequences, protein or nucleotide sequences between different organisms in its database. No, whether DNA or RNA or protein sequences. So imagine for our case, no, we have clean sequences, cleaned and assembled contigs. So yes, we have a sequences for my isolate, no, UPBCRE1. Now, what's going to happen is when we use that sequence, no, the sequence for UPBCRE1, and input it in BLAST, in Basic Local Alignment Search Tool, what will BLAST do is it will align our sequence, UPBCRE1, on a lot of sequences in its database. So what's going to happen is our sequence, UPBCRE1, will get to be aligned in all of the sequences here. I mean, on their database, on the NCBI database. And what's going to happen is it will create or will find all of those sequences that have high number of alignment, meaning it has a high number of similarity with our sequences. Therefore, if there is a high number of similarity, high number of alignment, it means that that sequence looks like our sequence. So it means that is the identity or the species of our or my isolates. So basically, that's the crux of using basic local alignment search tool. So if you think about it, it works like your NBI clearance, wherein you go to the NBI, and what will the NBI personnel will do is to find hits for that person, no, on their database. So it works something like that. Moving on, after going to that website, scroll down and go to nucleotide blast. So click it, and it will redirect you to this interface. So what you need to do is you need to choose file and find that Contig assembly, no, yung inedit natin. So one final Contig assembly, and we'll use UPB CRE1. And then we are going to input some parameters. Click RNA ITS database, and drop down, click the internal transcribe region ITS for fungi, since we are dealing with fungi. There's also for 16S for the case of your bacteria, but now we're going to use the ITS for fungi. And click uncultured to exclude, and remember sequences from type material. So you need to tick that and tick your highly similar sequences. Okay? And then click blast. And it will redirect you to this interface. So you just need to wait for it for some time. And then voila, here is the identity of your isolate of UPBCRE1. So go down and look at this portion. No? So what we're going to do is we'll first observe it. No? So as you can see, there are a lot of hits, no? A lot of sequences that match our sequence. And what you need to take note, and you will use this in your write-up, is the description, no? The identity of your organism, the accession number, the query cover, the e-value, and percent identity. So please pay close attention to this interface, specifically and particularly to the percent identity, because the higher the percent identity of our isolate to our sequence to these reference sequences in the database, the closer or the more alignment or the more hits there are in their sequences, no? The more similar their sequences are. And usually we only accept 90% and above, no, percent identity. And some actually uses only 97 and above, no, percent identity. Now, to give you an idea of how it should look like in your paper, no, let's look at my paper. So, what I did. So it looks something like this. No, we have your description, no, of your fungal isolates, what I named them, from where site, and you don't need to consider what site. You just put in the name of your fungal isolates. No, UPBCRE, whatever CRE is the name of it, and you need to put in the closest match with the accession number, the percent identity, and also include the query cover. No, it wouldn't hurt. Just tabulate it. Okay. Just to recap what happened. So for the part 2, we pasted the FASTA sequences in the blast platform, and then we hit blast, and we take note of the resulting interface, now the percent identity, query cover, identity of the organism. And because we will use this in the paper, no, we need to include this in the paper. And also, what should be included in your paper is the text file, no, of your cleaned and assembled config. So how should this look like? It should look like wait. It should look like, let me just open the appendix of my paper. So it should look like let me just find my appendix. So yes, here. it should look like this. Okay? So ayan, with the proper figures and figure labels and your caption for that figure. So it should look like this, okay? So how will you use that? How will you find that? Or how can you find this text file? So what you need to do is just find the config assembly for that file, no? The final config assembly. You put in Mega and you analyze UPBCRE1. Here it is. You can copy it and paste it in your Word document, just like that, just like so. And don't forget to put in the figure label. Okay, so that's how you're going to do it. All right. So moving on, let's go back to the PDF. Okay, here. So aside from that text file, we need the tabulated blast search result. No, so just like this. Okay, we need to include the percent identity, accession number, e-value, and query cover, plus the description or the name of your organism. Okay? Andito pa rin din siya sa baba. No? Ayan. Okay? So, accession number, so on and so forth. And then you need to have a short discussion of your result plus your literature cited. And after that, you can now move on to part 3. Oh yeah, before I forget, we need to right-click this accession number, open a new tab. The top three hits for your organism, for your sequence, and what we need to do is download the sequence file for those top 3 organisms. Okay, so how to download that? Click send to the drop-down portion, file, and FASTA format, and it will already download. Do it for the other top 3, no? And create file, it will download. You can now close this, send to drop down file, FASTA format, create file, and it will now download. Okay, so that's how you do it, no? And we do it for this number UPBCRE, the internal transcribe spacer, the same parameters, hit blast. We get to this interface, waiting interface again, and then just a couple of seconds here. Okay, we need to click open a new tab, those accession number, the top 3 again, and drop down send to file, FASTA format, create file. Okay, the same for all of them. Okay, all right. So you need to do that for all of the files, no, the sequence files that you have.
One eternity later.
Okay, after doing that for all of the sequences that you have, just remember, download the top 3 FASTA sequences format. Okay, so just a little tip, put them in a nice folder just like this. Number them if you want so you don't get confused which is which, okay? Now you can move on to the next part of the activity, part 3. So part 3 is Mega 10 phylogenetic tree construction. We're now going to create the tree for our sequences. Now, let me just, okay, we're now going to, first of all, we need to create the outgroup, download the outgroup. So go to this website again. So go to NCBI, and you need to, so you will be redirected to this interface. Click nucleotide, okay? So click nucleotide here. So it will redirect you here, and you need to input this exact words. Okay, 16S R RNA E. coli. So E. coli is a bacteria. No, it's not a fungi. So we're gonna use it as our outgroup. Okay, so click that, and just like the same for the others, file drop down, FASTA format, create file. And voila, there's the download. And just close this. Let me minimize this. Okay, so we have now an outgroup. And go to downloads. Let's just move it in the desktop.
One eternity later.
So just like the same for all of them, I created a folder for the outgroup. So we now have all of the things that we're going to use in our tree. So the final contigs that we assembled, the reference sequences from NCBI, and your outgroup. Okay, so yep, those are the three important things that we are going to use for the alignment in our tree creation. No? So you need to remember the clean sequences, the top three aligned sequences, and the outgroup. Okay, so all of these are what we're going to use. Okay, so how are we going to do that? So with those three file folders, we need to create an alignment. Okay, we need to create a new alignment file using Mega. Okay, so let me just minimize that. Open Mega, click Align, edit build alignment, create a new alignment, click OK, DNA, and let me just edit, insert sequence from file, find those three. So it's in my desktop. So number one, the final config assembly. You can control all or select them all, and let's insert the other one, the other folder. So where is it? Desktop. It's in my desktop. Okay, number two, all the reference sequences from the top three aligned sequences for your files. And the last one would be, so as you can see, here are the continued config assembly, no, for UPBCRE to 1 to 5. And we need the outgroup. So there's the outgroup. Um, yes, the outgroup. So your sequence for E. coli. So there it is. Okay. So okay, let me just put it in the middle portion. So it should look like this. Okay, a lot of sequences in one file in your Mega. Okay, it looks so cool, right? So let me just delete this blank sequence. Okay, and look at it, and you can rearrange them alphabetically. So yeah, that's how they look. Okay. Dun, dun. So we have the five. Okay. We have the UPBCRE 1 to UPBCRE 5, no? The 5 config assembly, and we have the reference sequences 2 plus the outgroup here. Okay. So these are your config assembly. Okay. So that's let me just, yeah. So as you can see side by side, we have, these are your clean sequences, no, from and config assembly from the last activity, and this is your, well, the top three aligned sequences from the blast search, no, yung dinownload natin kay NCBI din, yung top 3 na nag-align dun sa mga sequences natin. And of course, this is for your, ayan. So, siya naman yung kay outgroup. Yung 16S ni E. coli naman. Okay? So, okay. Let me close this na. Okay, so now what we need to do is select all of them. So, let's select all of them. So, you can control shift. And we use muscle alignment, align DNA. Just click OK. And just wait for some time. It's ongoing, aligning things, so much sequences. And here it is now. It should look like this now. Okay? It looks cooler now. Okay? So look at this. It has aligned the sequences somewhat perfectly, no? So you can use cluster and muscle alignment. It's okay. Those are algorithms that you can use, no, to align your sequences. So it should look like this. So, yep. So if it doesn't look like this, if it's all black, all blank, I mean, repeat the step. Okay? It should somehow look like this. And after this, we need to save this, export alignment into Mega format, and I'm gonna name this alignment alignment. I'm spelling align. Ano ba yan? Alignment. Yes. And just click OK. And click yes. Okay. You can now close this. So let me now just close this, and where it is, here it is, the alignment file. And what's next is we need to create the tree file, no? The tree. So Bootstrap tree file. So how to do that? Go to Mega again, and there are a lot of buttons. Click phylogeny. And there are a lot of tree methods, no, tree making methods to choose from. That I like to use the neighbor joining method. So in neighbor joining method, you need to wait lang, find your file. So yes, your mega file, no, alignment file, and you need to pay close attention to the bootstrap method and input 1000 bootstrap replication, and just follow all of this and then click OK. So just wait for some time, and there is your tree file. So it should look something like this. Okay, your tree file. Okay, so your tree file should look like this. Now, to cite how your tree was created by neighbor joining method, you can use this portion below, and these are the authors of the neighbor joining method of how it is created here. So Saito, Felsen, Tamura, and Kumar. Okay, so you can use that in your references. Just format it into APA, and as you can see here, this is the original tree. Now we're going to click the Bootstrap consensus tree. Okay, so click Bootstrap consensus tree, and as you can see, there are the Bootstrap number. So this is your tree. You can now export this. So how do you interpret this tree, no? Remember, we have set the Bootstrap replicate value to a 1000 replicates. So what does that mean? How do we interpret that, no? So sometimes, no, confidence estimates on the tree, well, to put it simply, interpreting the exact meaning of these bootstrap values, these confidence values in the tree, is still an area of debate, no? But experts generally agree that we can accept branches, no, with bootstrap support of 80% to 90%, no. So we need at least 80% or 90% at above bootstrap support, provided that there's an appropriate evolutionary model for the estimation of your for the estimated phylogeny that you have created, no. So for you to have a grasp of the bootstrap value, so it's like a tree making process kasi. So imagine if you have a 50 bootstrap value, the 50 bootstrap support, like here, no, for your Mucor fusiformis and Mucor himalis. So what does that 50 mean? So in that 1000 replication, in that 1000 bootstrap replication, 50% of the time, no, these two group together. Okay? So remember that, no? When you have a 90 bootstrap replication, or yeah, so that's why we always go for the 90 and above, or 80 and above for some authors, because what does that mean is in that 1000 replication, you have 80% of the time those two clusters together. Okay? So that's basically it. So the other cool thing about this tree, this mega software, is you can create all the different trees, by clicking. You can explore this more, You can create a circular tree with the same bootstrap value. You can create this beautiful looking tree. You can create a let me just a so there's a circle. There's a straight, something like this. Oh wait, doesn't move. Wait, what's happening? Okay, yeah. So you can create something like this, a branching cladogram, or curve one, if you're up to that. So you can create a straight, you can create a rectangular, you can create a straight and curve, You can choose what to use, Ang cool nito, specifically this one. But I would like you to use the rectangular one for your paper, okay? Okay, so after that, you can save your result and you should just whatever name you want, tree file, and so on and so forth. You close the data. You can just close it now. So the last portion that we need to do is to export this tree. So how to do that? You can click on the image and copy to clipboard, and you can just now paste it in a Word document. So just like this, and it should look like here, no, an image, no. So we have UPBCRE 1 to 4 and to 5. So all of them are here. So you can just copy it to your clipboard just like that, or you can export it as BMP, PDF, PNG, and TIF files. No? So you can just click what format you like. You can name it, and first you need to input the location you want to save it to, no, and the file name. No, let's use a more 1, 2, 3, 4. Yeah, file name, file name. So yes, and you can click that, and it's now saved as a JPEG. It should now be saved as a JPEG on that location. So you can discard that. You can now close that, and okay. So after taking the Bootstrap file and the screenshot, no, you can now create your paper. So you need that tree file kasi for your paper, and these are the same as the other activity, no? So the other formats. So please be mindful of that. So it should look like this in your paper, but not exactly this, of course. You will be using a different sequences, and you should highlight, no, the important sequences, the UPBCREs, and label them properly and cite them properly. Okay, so it should be included in your citation. So it should be included in your references. Don't forget to use those in your paper. Okay, so now we're done with that. Thank you for listening, and see you on the next activity. This is Sir Patrick. Peace out.